Alright, so today we're going to be looking at topic 2.3, carbohydrates and lipids, which comes under the greater topic of topic 2, molecular biology. Today's video will focus on carbohydrates, and then in the following video we'll look at lipids. Now the point that comes from your syllabus is that monosaccharide monomers are linked together by condensation reactions to form disaccharide and polysaccharide polymers. But before we break down what this means, let's define what carbohydrates are. Now, the word carbohydrates, or specifically carbs, is often tossed around without real knowledge or understanding of what they are. So today we're going to try and understand the scientific basis behind what carbohydrates are. Now, if we break down the word carbohydrates, the first part of the word, carbo, alludes to containing carbon. And the second part of the word, hydrates, alludes to containing water or the atoms that make up water, which are hydrogen and oxygen. And so carbohydrates are made up of carbon, hydrogen, and oxygen. Now, carbohydrates can either be simple or complex. We can have simple monomers or complex polymers. So our monomers are our simple building blocks, where our polymers are our complex structures. So we combine monomers together to make our complex polymers. So when we refer to the beads on the string analogy, our beads would be like monomers, whereas our necklace would be the polymers, because we put those beads together to form the necklace. Same way we put monomers together to form polymers. Now, carbohydrate monomers are called monosaccharides. So mono meaning one, saccharide sort of sounds like sugar, so one sugar. So those are the monomers, or the simple building blocks. And examples of monosaccharides are glucose, fructose, galactose, and ribose, some of which I'm sure you've heard before. When we talk about monosaccharides, they always exist in a fixed ratio, as in the carbons, the hydrogens, and the oxygens in the monosaccharides exist in a fixed ratio. And that ratio is a 1 to 2 to 1 ratio. So for example, if we take the molecular formula of glucose, which is C6H12O6, we can see that the ratio of C to H to O is a 6 to 12 to 6 ratio. And if we simplify this, this is a 1 to 2 to 1 ratio. So in monosaccharides, the ratio between carbon, hydrogen, and oxygen is a 1 to 2 to 1 ratio. Now, we need to be able to draw glucose. So this is what we're going to do and learn how to do on the following slides. We need to remember, just as was previously stated, that the molecular formula of glucose is C6H12O6. And this can help guide us as we draw to make sure we don't miss out on including everything that is needed in our glucose molecule. So, glucose is a 6-carbon sugar. We get this because in the molecular formula it says C6. So there are 6 carbons that are involved in glucose, and therefore we draw a 6-sided shape, a hexagon. The next step is to put an oxygen in the top right position of the hexagon. That can be a nice landmark to start. We fill in all the other spaces with carbons, and when we want to count our carbons, we start by looking at the carbon after the oxygen. So this would be our first carbon, or carbon 1. This would be carbon 2. This would be carbon 3, 4, 5. So this is how we number our carbons. And we can see here that we only have 5 carbons. And so we need to add one more. And we add that in here. So this is our carbon 6. So counting, remember, we start from the carbon after the oxygen. So carbon 1, 2, 3, 4, 5, all the way to carbon 6. The next step is we add hydrogens and OHs, or hydroxyl groups, to our carbons. And we add these to all our carbons except carbons 3, 5, and 6. So we put a H above the carbon and an OH below the carbon for carbon 1, 2, and 4. So essentially all the carbons except 3, 5, and 6. Remember, we put a H on the top, an OH on the bottom, a H on the top, OH on the bottom, and same here, H on the top and OH on the bottom. And once again, this is for all our carbons except carbons 3, 5, and 6. Then we add an OH and a H to carbon 3. But the difference is that our OH instead of our H is on the top. and then our H is on the bottom instead of our OH. So it's flipped around for carbon 3. So for carbons 1, 2, and 4, it's the H on top, OH on the bottom. For carbon 3, it's OH on the top, H on the bottom. For carbon 6, we add H2OH, and then we can count and see what is remaining for us to add. So we know that we need 6 carbons. If we count them, it's 1, 2, 3, 4, 5, 6. So we're complete there. With our oxygens, we need 6. 1, 2, 3, 4, 5, 6. And for hydrogen, we need 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So we only have 11 hydrogens, we're missing 1. And we put this hydrogen on carbon 5. So this completes our glucose molecule, and we can double check to make sure that we have 6 carbons, 12 hydrogens, and 6 oxygens. Here is a video drawing a glucose molecule. Try and see if you can follow along at home. Have a look at your glucose molecule. Does it look like this? Double check that it matches up with the molecular formula C6H12O6. Now, we've had a look at monosaccharides, but we've talked about how monosaccharides can link together to form disaccharides and polysaccharides. So glucose is an example of a monosaccharide. Other examples include fructose and galactose. When 2 monosaccharides join, they form a disaccharide. When many monosaccharides join, they form a polysaccharide. So mono means 1, di means 2, poly means many. So where we have 2 monosaccharides join, we form a disaccharide, where if we have more than 2 monosaccharides join, they form a polysaccharide. For each linkage made, water is produced. So the linking of monosaccharides together is referred to as a condensation reaction, because condensation reactions refer to reactions where water is produced. Thinking back to some knowledge that we should already have, is the linking together of monosaccharides to make di and polysaccharides an anabolic reaction or a catabolic reaction? Pause this video if you need time to think. Hopefully you said that the linking together of monosaccharides is an anabolic reaction, because it's a reaction where we are building up. One way that I like to remember this is that there are a type of steroids called anabolic steroids that some weightlifters use to build muscle, and so anabolic refers to building up. Another way that I've seen people remember this is by saying anabolic, ana builds. This helps remember that anabolic reactions are reactions where we're building up. So we take something simple, we take a monomer, and we build it up to make a polymer. So let's have a look at the disaccharides. 3 common disaccharides are formed when glucose is combined with another monosaccharide. So if we combine glucose with itself, glucose, we form the disaccharide maltose. Maltose is a sugar that is commonly found in wheat and barley. If we combine glucose with fructose, we get the disaccharide sucrose, which is commonly known as table sugar, or the sugar that we add to our teas and coffees. If we combine glucose with galactose, the disaccharide that we get is lactose, and lactose is found in cow's milk and other dairy products. So these are the 3 common disaccharides, and the linking together of glucose with glucose, or glucose with fructose, or glucose with galactose, or any monosaccharides together for that fact, is going to be a condensation reaction. So for each bond formed, we're going to have water produced as a byproduct. Let's have a go at drawing out the condensation reaction of glucose with glucose, forming maltose. So we know how to draw glucose, and that will be our first step. And we'll draw up another glucose molecule because that's what we're linking together. Then, the next thing we need to know is that the link takes place between our carbon 1 and our carbon 4. Remember, we've learned how to count our carbons. We start from the oxygen and look at the carbon following. So this is carbon 1. We do the same for this molecule. So this is carbon 1, carbon 2, carbon 3, carbon 4, and the link takes place between carbon 1 and carbon 4. We know that this is a condensation reaction, so water needs to be produced. Water is H2O, so we need to take out 2 hydrogens and 1 oxygen. We take out an OH from the first glucose and a H from the other glucose. These will form a bond to make water. Next, we use this oxygen to make the link between our carbon 1 and our carbon 4. So we take that oxygen and use it to form the bridge between these carbons. Now this link, where oxygen links carbon and carbon in a sugar, is referred to as a glycosidic link or a glycosidic bond. So now here we have formed a maltose molecule and water. So glucose plus glucose equals maltose and water. So for every link formed, so we've got one link formed here, we've formed a water molecule. If we were linking up 3 glucoses together, we would have 2 links, and therefore we would have 2 water molecules formed. How about you give it a try? Try drawing out the condensation reaction of glucose with glucose to form maltose. Pause this video if you need. Now we've looked at disaccharides, let's quickly have a look at polysaccharides. So we need to understand both the structure and the function of a few different types of polysaccharides. So polysaccharides are the polymers, and they are made up of hundreds to thousands of monosaccharide monomers. So we link monosaccharides together to form our polysaccharides. There are 3 main types of polysaccharides that we need to have an understanding of: glycogen, starch, and cellulose. Glycogen is found in animals and it's found in humans, and starch and cellulose are found in plants. Now these polysaccharides are all made out of glucose, it's just glucose linked together. However, there are slight differences to their structure that makes a world of difference to their function. So glycogen will function differently from starch, which will function differently from cellulose. Let's have a look at glycogen to begin with. Glycogen is a form of mid-term energy storage in humans. So we know that glucose is what we use to break down to give us our energy. However, if we have excess glucose, our body links all that glucose together to form glycogen, and then glycogen can be stored in the body. So excess glucose gets bonded together to form glycogen. The type of glucose used in the formation of glycogen is a type of glucose called alpha glucose. Alpha glucose is the glucose that we've been drawing, where we look at carbon 1 and the hydroxyl or the OH is below that carbon rather than on top. So alpha glucose refers to when the OH is below carbon 1 compared to beta glucose, which we haven't discussed yet, but if we look at carbon 1, the OH is above. So alpha glucose is a type of glucose found in glycogen. Now glycogen is not just a straight chain of glucose, rather it is a highly branched structure. So this is an example of glycogen here, and we can see that it's very branched. It's not just a straight chain. Let's have a look at starch. Now starch is also a form of energy storage, however it is not found in humans, it is found in plants. The type of glucose found in starch is also alpha glucose, so there is a similarity there with starch and glycogen. And there are 2 types of starch, amylose and amylopectin. Amylose is unbranched, whereas amylopectin is branched. So if we have a look at this image here, this would be amylose and this would be amylopectin. So amylopectin is branched, whereas amylose has no branches, it just forms this helix or this ring shape. Because amylose is unbranched, it takes up less space and can be packed more tightly compared to amylopectin. So amylopectin is branched and so cannot be nicely and neatly stacked, whereas amylose is unbranched and so it can be tightly packed for more energy storage. Amylose is also less soluble in water. Amylose is the more preferred form of storage in plants, and this is because it is less likely to be dissolved or destroyed by water, and also because it can be tightly packed compared to amylopectin. So remember, amylose is unbranched, whereas amylopectin is branched. So amylopectin is similar to glycogen because they're both branched. However, glycogen is more branched than amylopectin. Can you also state another difference between amylopectin and glycogen? Hopefully you said that amylopectin is a form of energy storage in plants, whereas glycogen is a form of energy storage in humans or animals. Lastly, let's have a look at cellulose. Cellulose is also a type of polysaccharide found in plants. However, it is not used for energy, but it is used for structure and formation. And it is found in the cell wall of plants and gives plant cells their rigid structure. It is linear and unbranched, and this allows for it to be stacked nicely on top of each other to provide that rigid structure. The type of glucose found in cellulose is beta glucose. So if we think back to our image here, beta glucose is where the hydroxyl is above carbon 1 rather than below carbon 1. And so most animals lack the enzymes required to break down beta glucose, which is why cellulose is indigestible to most animals. And actually, when we're talking about fiber, we're referring to foods that are high in cellulose because fiber is unable to be digested, and that is due to it being made of beta glucose. So that's a difference there. All our other types of polysaccharides are made of alpha glucose, whereas cellulose is made of beta glucose. Here's a nice table from BioNinja that compares these polysaccharides. So we have cellulose, starch, and glycogen, and then starch can be further broken down into amylose and amylopectin. We know that cellulose and starch are found in plants, whereas glycogen is found in humans. For all of them, they're made of alpha glucose, except for cellulose, which is made of beta glucose. Now we remember that glycogen is branched, and so is amylopectin, but amylose and cellulose are unbranched. And this is what these 1,4 and 1,6 links mean. So a 1,4 link refers to being unbranched, whereas the 1,6 linkages is where the branches take place. So if we look at this diagram here, this explains it nicely. So remember, the link takes place between carbon 1 and carbon 4. So when we count from the carbon after our oxygen. So this would be oxygen, this would be carbon 1, this would be carbon 4. So this is a 1,4 link. Here this would be carbon 1, this would be carbon 4. So this is a 1,4 link. Carbon 1, carbon 4, 1,4 link. We can see that there are 1,4 links here as well, but there's also this link going on. So if we count our carbons, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, this rectangle here representing carbon 6, we can see that there is a link here between carbon 1 and carbon 6. And so that's what we refer to when we're talking about that 1,6 link or 1,6 bond. That refers to the branches. So for our things that are branched, they contain 1,4 and 1,6 bonds, whereas for our things that are unbranched, they just contain 1,4 bonds. And once again, here is a comparison of the shapes: highly branched, bit less branched, unbranched but helical, unbranched and straight. That's all for this video. Happy studying.